Welcome to the next video in the evolution series. This video will be looking at 2 dot points from the Life on Earth syllabus, 8.4.32, which is describe the main features of the environment of an organism from one of the following groups and identify its role in that environment. And 8.4.3a, use the available evidence to outline similarities in the environments past and present for a group of organisms within one of the following. So the 2 groups of organisms that you need to choose from are Archaea and Bacteria. Now this video is going to look at both of them, as I don't think it's a bad idea to have a bit of an understanding of both and be able to compare and contrast, but you need to choose one that you understand in a good amount of detail. So let's start off by having a look at the Archaea. So Archaea are prokaryotes without peptidoglycan in their cell walls. So peptidoglycan is a polymer, a sugar polymer, which helps to create a membrane around the cell membrane of the organism. Archaea are unique and are thought to be very rare, so they're not found in as many places and in as large abundance as normal bacteria. And it is possible that they are relics of the earliest forms of bacterial life. So the bacteria that we know today is believed to have formed from these organisms in the past. Evidence to support this comes from their anaerobic nature and their ability to survive in extreme environments such as swamps, near boiling hot springs, salt marshes, and the digestive tracts of herbivores. So we recall that anaerobic means that they can create energy without needing oxygen. And the interesting thing here is that they're found in a lot of environments where normally other organisms wouldn't be able to survive. They also carry out an unusual metabolic pathway, so very different to the other organisms that we know on Earth today. So what we're going to do is have a look at the different groups of Archaea. So as we know, there are ancient prokaryotes. So the first one is the methanogens. So from the name, we can see that they have something to do with methane. So what these Archaea do is they produce methane as a byproduct of metabolism. So we produce carbon dioxide as a byproduct of metabolism. They produce methane. They're the largest group of Archaea, so there's more of them than any other group. They are anaerobic, so they don't use oxygen as an energy source, and they're found in areas where there's little or no oxygen. So they're found deep within the soil, in swamp mud, sewage treatment plants, and in the digestive tracts of some animals. The next group we're going to look at are the halophiles. So these ones also don't use chlorophyll, but they use a substance called bacteriorhodopsin for photosynthesis. They're thought to be aerobic, so they do possibly use oxygen as an energy source, and they live in places with high salt concentration. Okay, so there are groups of plants called halophytes, which are plants that are able to deal with high salt concentration. So that prefix halo just refers to high salt concentrations. The last group we're going to look at is the thermophiles. So as you can tell from their name, thermo obviously referring to heat. These guys live in hot places such as near boiling springs. So in places like in Yosemite in America, those geothermal hot springs, also in New Zealand. They found in areas of volcanic activity where temperatures are high, so from 50 to 100 degrees Celsius, where a lot of other organisms would not be able to survive for long periods of time. Deep sea bacteria thrive near the vents along the mid-ocean ridges, and sulfur is produced, the sulfur produced by the vents is used as an energy source. So again, they're not aerobic, they're anaerobic, they use sulfur to create their energy. So let's have a look at Eubacteria. So Eubacteria, or simply bacteria. So these are known as true bacteria. So remember when we looked at eukaryotes versus prokaryotes, Eubacteria, true bacteria, just like eukaryotic meant true nucleus. So these organisms thrive in most environments and can live in the air, soil, and the water. They're often about 1/10 of the size of a normal eukaryotic cell, so a normal plant or animal cell, so they're extremely small, and they can be either rod-shaped, so a spherical shape, or spiral in shape. So depending on the type of bacteria that it is, they have a definite shape that allows them to be identified quite easily. So just like we looked at the different types of Archaea bacteria, we're now going to look at the different types of Eubacteria. So the first group we're going to look at is the nitrogen-fixing bacteria. So these guys are found in the soil amongst the roots of plants. They play an important role in the nitrogen cycle. Obviously, from their name, we figure that they have to have some kind of role in something to do with nitrogen. So nitrogen fixation is the process where these bacteria are able to transform atmospheric nitrogen into usable nitrogen compounds. So they take the nitrogen out of the atmosphere and convert it into ammonium, which is more useful to us. The next group are our disease-causing bacteria, and these are probably the ones that when we think of bacteria, we think of most commonly. Now, disease-causing bacteria are found everywhere. They're found in the soil, in the water. They can be transmitted through the air. They can be found on surfaces, and they can also be found in the digestive tracts of animals. So a couple of diseases that are caused by bacteria include diphtheria, tuberculosis, tetanus, and syphilis, all quite unpleasant and diseases that you probably wouldn't really want to get. The big thing with disease-causing bacteria is they are sensitive to chemicals called antibiotics. So antibiotics can kill bacteria in a number of different ways. And when we move on to the Search for Better Health topic in Year 12, we'll be spending quite a bit of time looking at bacteria and antibiotics, how antibiotics are created and how they work to kill different bacteria. And the last group of Eubacteria are the cyanobacteria. We've mentioned these a few times throughout this topic so far, and we know that they contain chlorophyll and therefore are photosynthetic. And as we know, we've talked about, these were the first ones that started to produce the free oxygen that started the real cycle of evolution. They can live in colonies or singularly, and they are found in stromatolites dated to be about 3.45 billion years old. So the earliest evidence of cyanobacteria is 3.45 billion years old in stromatolites that have been found in Western Australia. So let's have a look at the past environments of one particular group of Archaea bacteria, being our Thermoacidophiles. So remember, these are the ones that live in very hot environments. Now, thermophiles live in very hot environments. Acidophiles live in an acidic environment. So thermoacidophiles are those Archaea bacteria that live in environments that are extremely hot and also are quite acidic. So fossils have been found of these thermoacidic phials dating back to 2.75 billion years ago. During this time, the Earth was in the early stages of formation, so temperature would have been extremely high. All Archaea display properties of autotropism, which means that they're able to make food using the materials present in the Earth's crust. In this case, back in early Earth, there was hydrogen, sulfur, and carbon dioxide. The conditions present during the early stages of Earth favored Archaea over photosynthesizing or heterotrophic organisms. So the oldest fossils found in mines of Ontario in the Canada, which is an area believed to have been submerged and surrounded by hydrothermal vents. So remember, we said hydrothermal vents give off temperatures between about 50 to 100 degrees Celsius. There would have been no direct sunlight or oxygen, and as we've already said, extremely high temperatures. Thermophiles do not require oxygen to survive, thus they are believed to have existed during the early formations of Earth when there was little or no atmosphere present. So what about present environments of thermoacidophiles? So recently, these organisms have been found in places such as geysers, where extremely high temperatures can also be found. They've also been found in hot lakes and springs, such as the hot springs of the Yellowstone National Park in the USA. They've also been found in locations that are anaerobic and extremely acidic, so a pH of less than 5, as well as having the high temperatures. The scientists believe they may be one of the last common ancestors of all current organisms and species present on Earth. Similar environments to the past exist in places like Iceland, where scientists recently found the first ammonia-oxidizing extremophiles in acidic hot springs. The environment is hot, acidic, anaerobic, and shut out from the open world, just like the mines in Ontario where they found the other fossils. Vents located on the ocean floor do not receive any sunlight, and there is little or no oxygen present, and these conditions are similar to those in the past, allowing these thermophiles to survive. So what we've done is we've had a look at a couple of different groups of both Archaea and Eubacteria and where they live and what they do, and then we've compared past environments with present environments to look at the similarities, specifically for Thermoacidophiles, which fall under the Archaea group. And that brings us to the end of this video. Thank you for watching.